Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the video is related to RNA sequencing data analysis, and the title of my today's video is Understanding FASTQC report. It will be an end-to-end explanation. As you all know that I have started a video series related to RNA sequencing data analysis, and this is the part 11th of that video series. In today's video, it's a bit theoretical, and we will cover these concepts. Basically, these are the reports of FASTQC. I will discuss the basic statistics per base sequence quality, per tile sequence quality, per sequence quality, and other else. So stay tuned till end because it's very much important. And because when you understand FASTQC reports, so after that, then you can easily pre-process your data using different pre-processing tools. Okay, so if you understand it well, so you can pre-process your data well. Okay. So in the previous video, in the part 10 of this video series, I perform FASTQC on our files, and I practically show you that how to perform it. So if you haven't watched this video, so I recommend to watch this video because you should know that how to perform FASTQC using Linux operating system. And after that, when you are familiar with it, first perform your FASTQC on your biological data on your sequencing files. After that, then interpret it. So today is basically the interpretation of it. Okay, before proceeding to the video or to the explanation, I want to tell you that in the part 4 and part 5 of this video series, I briefly discussed the structure and quality of sequencing files, and I also told you that what are the basic reasons that make our files compromised, okay, with respect to quality. So I recommend to watch part 4 and part 5 of this video series. So it will give you a brief background about the quality of sequencing files. After that, then watch this part 10 and then proceed this with part 11. Okay, so the only reason is that you will be in a flow and you will not miss even a single point of it. Okay, so I recommend to watch that videos, then start this video. Okay, so basically now we will start with the FASTQC reports. FASTQC reports have basically three type of colors. One is green color, that means passed. The second is warning, and the third is failed. Okay, so green, yellow, red. So basically when it is green, so it is okay, and after that when it is warning, so it is also okay, although you can perform pre-processing to just clear that warning and make it pass. But if it is not if you are not worrying about it, so warning is also okay, you can proceed with your data. But if there is failed, so then you have to pre-process your data according to the understanding. Okay, so the main reason of this video is to make you understand that what basically these FASTQC reports tell us about. So when you interpret it well, then you can easily pre-process it and remove the failed or the warning regions, okay, and make it green. Okay, then because this is very much important. Okay, so the first stat is the basic statistics. Okay, and basically it is always green. So basically it show you the detail of your file. For example, my file name was SRR7217471 and it is of first read, paired and read, and it is a fastq.gz. So this is my file name, and my file type is conventional base calls, or basically it is a sequencing file. Okay, fastq file. After that, it was being generated by Sanger Illumina 1.9, and the total sequences are 48,34,426. So the total reads are the total sequences. Now the total bases are 557.9 million base pairs. Okay, and after that, the base pairs are basically the adenine, guanine, cytosine, and thymine. After that, sequence flag is poor quality. So basically, it does not have any poor quality. Okay, so when you are dealing with the raw files or the raw data, so basically it should contain some poor quality. Or if it is pre-processed already from the company, so then it will there will be no poor quality. Okay, so basically it shows the poor quality. Now the most important thing is the sequence length. So basically my sequence length here is, it starts from 60. The lowest sequence read is 60 and the largest is 150. Okay, so basically it was I pre-processed this data, and this is the FASTQC report of that pre-processing was. So basically this is the report of it, and the number of the the percentage of the GC containers is 49%. So basically it should be 50%, 49%, it's okay. Okay, but so this is all the statistics of my my FASTQC report, and it is always green. So now we will proceed with the per base sequence quality. So before proceeding with it, a very small concept that I already mentioned in the previous videos, but here I'm also giving you a very short introduction of it. Basically a FASTQ file consists of sequencing reads, and inside that FASTQ files there is a header, there is a, in the second line, this is our sequence, the sequence read, this is the read, and after that there is a spacer, and in the fourth line each it is the quality of each base. Okay, so for example, the quality of this first C is basically this under rate. Okay, so it shows something about the quality, and basically it just follow these ASCII rules. Okay, so basically when there is this under rate, so this means that it has the probe of error is 1%. So I already have discussed it in the previous video, but it just to remind you, because we will be dealing with this result. Okay, so basically the first is the per base sequence quality and how to interpret this. Okay, because it is very much important. It consists of three regions: the green region, the warning region, and the error region. Okay, so if our bases are in the error region, so it will give this red region, so it will give an error. Okay, so I will explain these are the box plot of it. Basically this is the position in read base pair. On the y-axis is the quality, and basically this is the quality. These are the quality. Okay, the Fred score is the quality, and after that this it starts from the 0 and ended with 30. Okay, so 30 is the highest quality. Okay, so on the y-axis is the quality, and on the x-axis is the number of base pairs. Okay, so basically I will explain a single of it. For example, we are looking into the quality of the first base. Okay, these are my sequence reads. Okay, these are my sequence reads, and this is the quality of that sequence read. It's a type of arbitrary, but I won't explain it. I want to show you that how the FASTQ just read it. Okay, so I will be dealing with the first box plot. So for the first box plot, it just revolve around the first base pair. Okay, so basically we are having 32 base pairs. Okay, if we count it, so it is 32. So it basically gives us the 32, the quality of the 32 base pairs. Okay, so after that what we'll do, these are my first base pair, and I want to check the quality of it. Okay, so it will be shown in a type of box plot. Okay, and this is the box plot, and this is the first base pair. So collectively it represent its quality in a type of box plot. I think you all are familiar with the box plot. It just show you a range of quality. So for example, in this box plot, it have a range of quality of this yellow color box also shows a specific percentage or specific amount of quality and amount of read that lies in this quality. And this is the whisker. Okay, so basically we can say that about more than 50% of our data is present in this region. Okay, so for example, the first four bases, the first four bases of the first four sequence reads are their quality lies in this range. Okay, so basically it shows you that the quality of the first reads are in between this range, and for example this is 32 and 33. Okay, and the last one we can say that its range lies in between 29 and 32. Okay, so basically we can say that overall the quality of my first base is almost good. It lies in between the 29 and 33 of this. So basically it is present in the green region. Okay, so you can also interpret just like this the other part also. Okay, so this blue color is the median, and this red color is the median, and this blue color is also to something I will now show you. Okay, so that is how FastQC just check our reads, and it give report of each and every base. Okay, collectively. So the first base, quality of the first base in every read, basically it can be represented from a box plot. Okay, so I think you now get the point. So for each read it is collected. Just look here to the 32 this 29th read. Okay, for example, so for example 29th read has the very much lowest quality. Each base has a lower quality. Okay, so basically most of the bases have lower quality, so it rise in the error region, almost in the error region. Okay, so that's how it is being calculated. Okay, so basically the central red line is the median value, and these are the yellow box represent the interquartile range, 25 to 75 percentile. So basically what I was telling is that here is about 50% of your data is here. Okay, so about 50% of our sequences have good quality. Okay, the first base, 50% of the bases of the first base have good quality. Okay, and that's what I mean. The upper and the lower whisker represent the 10th and the 90th percentile. Okay, so basically here this is the whisker. These are the whiskers. Okay, but here it consists of an upper whisker and lower whisker, but here almost all the quality is revolving inside here. Okay, so most of the quality is here on this red line. Okay, so no whisker is being present. Okay, if there was a sequence read and its quality was 34, so then there would be a whisker. Okay, but here almost all the quality revolve around here, this 33. Okay, so that's why it does not have a whisker. And after that, this whisker is a little bit larger, so we can say that a couple of reads have a quality in between 32 and 29. Okay, so that's why. So there should be a whisker and there should not be a whisker. If here is not the whisker, so we can say that almost all the reads, the 25% of the reads revolve around this median. Okay, so that's why. So this is the reason that here is not the whisker. And the blue line represent the mean quality. Okay, so this is basically the mean quality of it. So hope you get my point. And if you know that how to interpret it, then now here that our 20 from 28 to 32 reads have compromised quality. So then now you can deal with it. And I will show you that how to deal with it in the later video. Okay, in the part 12 of this video series. Here I just want you to understand the results of it. Okay. Now, we will proceed with the per sequence quality score. So it's very much easy. Basically, it just check our sequences. Okay. Basically, our sequences were. I want to show you that in the statistics, our sequences were 48 lakh 34,426. So basically, almost this is the quality, the fret score, and this is the number of sequences. So it start from the 5 lakh and it just go to the 25 lakh. I think so. Yeah, to 25 lakh. So almost 25 lakh or more than 25 lakh of our sequencing reads have quality of 35, in between 31 and 35. Okay, the entire quality of the sequence read. Okay, so it means that we are having a good the sequences that are present in our FASTQ file have good quality. Okay, overall good quality. So it should be like this. Okay, so this is the per sequence quality score, and it should be the distribution of average read quality should be tight in the upper range of the plot. Okay, what about this is also a per sequence quality score. Now we are having some sequences that have a overall quality in between 10 and 20. Okay, and 10 and 20, in between 10 and 20 this is not a good quality. Okay, so about this is another report. About 15,000 of our sequencing reads have a quality in between 11 and 20. So this is an issue. Okay, you have to deal with it. And after that about 60,000 of our read have quality of 20 in between 24 and 33. Okay, so it's also good, but the perfect was this one. Okay, so you have to deal with this such type of things. Now the third one is the per base sequence content. Okay, so before proceeding with it, I want to show you that when we are having the read, so the nucleotide, the bases, the adenine, guanine, cytosine, and thymine all should be in equal number. Okay, if thymine is 4 times, so adenine should be 4 times. If cytosine should be if it is 4 times produced, so adenine guanine should be also 4 times. Okay, so basically all the bases should be in equal number. So what if there is an issue and our sequences, the bases are not in equal content? Okay, so basically then there is an issue, and this report is just show you like this, that 1 to 9 in between the 1 to 9 bases of the all sequencing read, it just check all the sequences read just like this, and after that it gives the report. So it check guanine, thymine, adenine, cytosine, and after that it check that either its number is equal or not. Okay, so just look here on the first position, the adenine, guanine, cytosine, thymine is not equal. Okay, just look here, thymine is red and cytosine, adenine, guanine is yellow. So the range is not equal. Up to 9th, or we can say up to 12th of the bases, the number are not equal, and I will show you that what is basically the reason behind it. And I also discuss it in the part 4 and part 5 of this video series, but I will also tell you here. So here the number of bases are not equal, although after the 15 base of the sequencing read, now all the bases are equal. Okay, so basically this shows the per base sequence content. Okay, so here why 1 to 9 is basically not equal. So almost in the part 2nd and part 3rd of this video series, I explain the sequencing technology and the adapters. Okay, so basically when we are sequencing our reads, so there are adapters in the beginning and in the end of our sequencing reads. So basically the adapters are human made sequences, and it is just meant to recognize our read. Okay, so basically, and to start the sequencing, so it is human read, it does not follow the number of bases and the equal content of the bases. It's just made because to just understand or just to identify the sequence. So it may consist of plenty of adenine, no cytosine or guanine. So basically when they are sequenced in your FASTQ file, when they are shown in your FASTQ file, so it will give you such type of result. And after that, what will you do? You have to remove the adapters and the other such type of content, the contamination and other type of content of it. After that, you will then see that this would not appear. Okay, so I will show you that how to pre-process it, but just keep the concept in your mind. Now, per sequence GC content. Okay, so as I have told you that GC content should also be about 50%, because if adenine, thymine is 50%, so GC content should be also 50%. Okay, so it also show you that what is the percentage of your GC content. So it consists of a theoretical distribution, and the GC content per read, it shows just like here. Okay, so you can interpret it if there is an issue. So this pink line, this dark pink line will be just elevate this dark blue color line. Okay, so 50% is important. Okay, or 40%. So I told you in the start of this video series when I was showing you the statistics, so my report shows that it was 49%, and it is good. Okay. Now, reports the percentage of n at each point. Okay, so this is per base n content. Before explaining this n content, in the previous videos, I show you that if we are having a sequence and there is an ambiguous call, this n is basically ambiguous call, and I have told you the reason in the previous videos that why these are being generated. So these are the ambiguous calls, and our sequencer does not know that what should I tell, that either it is adenine or it is thymine or cytosine or guanine. So basically, just to overcome or just to mention this ambiguity, it just give the sequence as n, that I do not know that I have, or I have sequenced it, but I was not sure about it, and what was basically this content. Okay, so these are the ambiguous calls. So ambiguous calls are also very much important to be removed from our sequencing reads. Okay, so per base sequence. Now here, if you are having no ambiguous reads, so basically sequences reads that are having no ambiguous calls, so basically this line will be just like this. Okay, so 1 to 150, there is no such n content. Okay, so when it is more than 5%, so basically it is a warning, and if it is more than 20%, then it is an error. Okay, so keep this thing in mind. Okay, now in the sequence length distribution. Okay, sequence length distribution is also very much important. In the start of this video, I show you that my sequence was in between 60 to 150. Okay, so the length of the sequencing reads also matter. So the maximum the length, the better is the, it is better that it should be near 150 or so. Most of our sequences, these are the number of sequencing reads, and most of the sequencing read have a length in between 150 and 151. Okay, although there are reads that have length distribution in between 58, 59, 64, 65, but it is less every number. So it is a perfect, not a perfect, but it is a warning, but it is better. Okay, so your sequence, it depends upon you when you are performing pre-processing. So you can tell your fastp tool that how much length do you require. So you can just make a threshold of it, that I want a sequence, those read that have a sequence length in between 100 and 150, or 140 and 150. It depends upon you. Okay, so you have to decide that how much length I require, but the basic length is almost from in between 70 and 150. Okay, so I recommend to you to choose 70 and 150 of it. Okay, now the sequence duplication level, it is very much important, because there should be no such duplication. There is a specific threshold of duplication, but if your entire sequence is repeated again and again, so then it is an issue. Okay, so duplication means when your entire specific read is produced, sequenced again and again, and the reason is the polymerase chain reaction. Before proceeding with the sequencing, or with the next generation sequencing, you just increase your number of reads with respect to PCR. Okay, so PCR just amplify the sequences, and it does not follow that the hard and fast rule that it should magnify or just amplify every read in a single time or two times. Okay, so it can amplify a read 100 times, and it can amplify a second read 10 times. Okay, so it depends upon the PCR of it. Okay, so when you perform PCR and you come upon with specific reads that are being amplified about 1,000 times, so then it will be sequenced in the sequencing, and after in the sequencer, and after then it will show you an issue. Okay, so then you should have to remove the duplication, because when you are doing alignment and gene count, so then it will show you that this gene count is highly expressed, although it was duplicated. Okay, so you have to remove the duplication of it. Okay, so just look here, about 10,000, more than 10,000 of a specific reads are being duplicated. Okay, so you have to remove the duplication. So percent of remaining if deduplicated. So there is also a range of duplication. Now here it is it does not follow the threshold, so basically here is an error. And in the pre-processing, there is an command that when we use it, so it removes the entire duplication of it. Okay, so but you have to understand what is duplication. Duplication is basically the repetition of entire read. Okay, for example, if you are having read 1 and it is being duplicated 10,000 times, okay, so it is an issue. Okay, so you have to resolve it. Now, overrepresented sequences. Okay, most of the people are get confused in between duplication and overrepresented sequences. Here I will clear it out. Okay, overrepresentative sequences are basically short sequences that are present in the entire read, and they are being produced overrepresented. Matlab, they are produced again and again. For example, if you are having 10 reads, and in that 10 reads, this specific region is repeated again and again. Okay, so then it is an overrepresentation. Okay, because the software will detect that it is not a type of thing that a specific sequence should be repeated again and again. It should have an issue. For example, if we are using primers, okay, sorry, the adapters, and when the adapters are a specific reads that are that are present with the sequence reads, okay, and with our sequence reads. And after that, when it is sequenced, okay, so adapters are present with every sequence, and it is man-made, so it is sequenced again and again, again and again in the read. So when the FASTQ just scan it, so it come upon a specific portion of a read that is being over-represented. It is present in each and every read, so it is not we can say that it should not be there. Okay, just look here, this the first portion, this is a sequence that is present in 66,35,000 counts. Okay, it is how this is, so this portion is over represented, so we have to remove it. Okay, because there is no such hard and fast rule that every gene should have a specific portion that is common in each and every part of a sequence. Okay, in every gene, okay, for example, if we are having 10 genes, so there is no such specific portion that is that is repeated in every gene. Okay, because the gene sequences are different from each other. Okay, so this is basically over representation and it should be removed. Okay, so it is just due to adapters or different contamination. Okay, so duplication versus over representation. The sequence duplication is basically the repeated occurrence of the entire reads, and over representation is basically repeated occurrence of a specific sequence or motif, okay, of a smaller portion of the read. Okay, so this here is the entire read when it is duplicated, and here is a portion of a sequence inside that read. Okay, and if it is repeated in each and every read, so then it is over representation. So I think that now you can just differentiate between duplication and over representation. Okay, you can read this content. After that, adapter content is the last part of this FASTQ reports, and basically I very much in detail I discuss the adapters, that what are basically adapters. So watch part third of this video series. I will also share the link in the description. You can just explore the concept of adapters. Okay, adapters are basically man-made just to recognize our sequences. Okay, so it should be removed. Okay, because it is not an actual read, an actual basically read, so it does not code anything. So when it is being present in our sequence, so it should tell you it should donate some issue. Okay, so it should be removed. Okay, so in my FASTQ report here there are no adapters. Okay, so basically it is green. So if adapter is present more than 5%, so it should be an warning, and if it is more than 10%, so basically it should have it should give an error. Okay, so this was about adapters. Okay, so I think I have tried my best to explain each and everything. Now when you just understand these results, I think I recommend to watch the previous videos. After that, watch this video. Okay, so then I'm pretty sure that you will be almost completely okay with the quality of the file. So after that, in the next part of this video series, I will show you that how to pre-process it. If for example you are having this such type of condition, okay, that most of your reads are being in the range of 28 and 32, and have a lower quality, so how to deal with it and how to clean your data. Okay, if you are dealing with this such type of thing, okay, so how to deal with this. Okay, so I will briefly show you the commands, okay, that how to pre-process it, and then just play around your data, download your data. I have shown you that how to download your data and pre-process it. Okay, and after that you can just clean up your reads. Okay, so this was all about the today's video. If you have any question, just do let me know. I will answer you in the comment box. You can also email me about your queries, and if you have any issue with your final year projects related to bioinformatics, so do contact me. I will help you out. Okay, so thank you very much for today. See you in the next part of this video series. Thank you very much. God bless you.